All right, so talking a little bit about transcription from current slide. Here we go. So what we're basically talking about when we're talking about how cells use their DNA to make complementary copies of mRNA and then make the amino acid chains that form proteins is step by step by step, where we have transcription, which is how the information gets from the DNA to the RNA, and then translation, which is how the amino acids get put in the right order to make the correct protein that we want. So here in transcription, as you can see in the upper left-hand corner, we've got our strand of DNA, and it's coiled tightly up, it's wound tightly up. And the first thing that's going to happen is the recognizing and binding. So RNA polymerase recognizes a promoter site, which is that spot at which the DNA has started to unwind. And the RNA begins to make a copy of the DNA. So DNA double-stranded, RNA is single-stranded, and it begins basically reading this gene message. So RNA is getting ready to make up this strand that's complementary to the DNA. So the second step here is transcribing. As the DNA strand is processed through the RNA polymerase builds a single-stranded copy of the gene called mRNA transcript. So what you see here is this big greenish glob, and this is RNA polymerase, which is a chemical that's helping to attach the pieces of mRNA to the DNA to make these complementary strands. And what it does is it slinks along and goes down the DNA and keeps adding in the pieces of mRNA to make this single-stranded copy of the gene. So the third step that we have in the upper right-hand corner is that when the RNA polymerase encounters a code signaling the end of the gene, so we have codes that signal where the mRNA is going to start copying. We have another set of code, like a sequence of bases of the DNA, that says where it's going to stop copying the gene. And so it stops copying and stops making the mRNA transcript. And so then we have this chunk of mRNA. And as you can see in the middle there, we've got the DNA is unwound, but only at a certain point, and the mRNA comes in, makes that complementary copy. And then as you can see in the middle, the mRNA, as it detaches from the DNA, it's going to have what's called a cap and what's called a tail. So the cap is the mRNA that's looking for this complementary code. It starts from there. And then the tail is the non-protein coding regions of mRNA. So capping and editing. Before the mRNA transcript can be translated into a protein, a cap and a tail are often added for protection and to promote recognition, and non-coding sections are removed. So we've got this really long strand of code. It's got a cap on one end and it's got a tail on the other end. And the cap basically makes it so that as it's moving throughout the nucleus and the nuclear pores, that it's not going to be destroyed from one end. And the tail is kind of this free-flowing tail. And sometimes little pieces of the tail fall off, or the RNA polymerase is going to chew it up and basically take pieces off of the end of it. And so this is a very temporary situation, and this happens over and over again, billions of times within the nucleus. So they have this awesome code. But imagine if during your recipe that you are making chocolate chip cookies, if you have 5 directions in order to make the cookies, the tail end of the mRNA would be like if we started erasing up from the bottom. At a certain point, you don't know how to make the cookies anymore. So you have to be able to read that recipe, and at some point they take away so much of the recipe that it's not readable anymore, and that's when everything starts to break apart. So we see the RNA polymerase, which is that chemical. It's moving down the strand of DNA, so it's unwinding it, and the helix unwinds so the DNA can be read. It's got to be read from an open spot. At the same time, the DNA that has already been transcribed, so the part that's already been read, rewinds itself back up into the double helix form. And so I think a lot of times students think that this happens at the end of the DNA, but it actually happens where it, imagine if you busted your zipper on your coat, right in the middle of your coat. Not from the end and not from the top, but if you bust it open right in the middle, that's exactly what's happening here. So this is one of the videos that he had wanted to show, and I'm not sure if it'll play in the browser. If it doesn't, I'm going to show you where it is on the site.
The transcription of a gene into a messenger.
Let me start it over again.
It went off on our side.
A structural gene is made up of a sequence of bases in a DNA molecule.
Oh. All right, let's listen to it. You know, this is so strange. Let me see if that works better on my computer. That's my Twitter. What's up with that?
I think you're supposed to use the speakers behind you.
The transcription of a gene into a messenger RNA sequence is initiated when RNA polymerase.
Well, how strange is that? He said he uses these speakers?
No, but if you plug in that green cord to your computer, that's right there.
Okay. Let's see.
DNA molecule until the RNA.
I think you cut it on top of the little thing.
RNA polymerase
there we
to
go.
dissociate from the DNA molecule, and the completed transcript is released.
All right, one more time.
A structural gene is made up of a sequence of bases in a DNA molecule consisting of a coding region with an upstream promoter and a terminator downstream of the coding region. The transcription of a gene into a messenger RNA sequence is initiated when RNA polymerase attaches to the promoter region and forms an open complex. RNA nucleotides are then polymerized from nucleotide triphosphates to make a messenger RNA. The messenger RNA sequence is elongated as the RNA polymerase moves down the DNA molecule until the RNA polymerase reaches the terminator region. Transcription is terminated when sequences in the terminator region are encountered. This causes RNA polymerase to dissociate from the DNA molecule, and the completed transcript is released.
Okay, so basically to recap what is happening here, you can see that the DNA has these long strands, and right there in the middle, it's green on both ends because those are the parts that are not being transcribed. And then in the middle, you've got the blue section, and that RNA polymerase, that chemical that comes along, and attaches on, and it begins unwinding right from the middle, and it's making that copy of the RNA. Once it reaches the terminator ends, it's going to say, all right, time for me to stop doing that, and it detaches from the DNA, and then the RNA polymerase goes on to its next strand of DNA and begins unwinding it. And so we've got our complementary strand of RNA to that DNA, and that's how the cell is able to read those pieces of DNA. So why don't I stop there for just a minute, and I'll let you guys take 2 minutes and